Hello everybody, welcome along to today's webinar. My name is Jamie Clayton. I'm an Operations Director at Freeman Technology, part of the Micromeritics Instrument Corporation, and I'll be moderating today's session. So first of all, thank you for taking the time to join us. Hope that you find today's presentation interesting and informative. As many of you will probably know, this is part of a series of webinars that we've been holding over recent months, so thank you if you've attended previous events, but also please keep your eye on your inbox and on our website for details of upcoming events, including some great guest speakers. Before we get started, just a couple of housekeeping things for today's event. So you'll notice when you joined that all attendees are in listen-only mode. So if you do want to raise a question, you can click on the speech bubble or the question mark in the GoToWebinar dashboard, and you can submit your question there. When we get to the end of the presentation, we'll hopefully have time to address some of the questions, but rest assured that if you do ask something today that we don't get a chance to address, then one of our application specialists will come back to you in the coming days. Please note as well that any questions are just visible to organisers, so they won't be shared with any other attendees. One other quick word of caution, we have had some reports with the GoToWebinar software that sometimes the audio can drop out on the transition from presenters. If this does happen to you today, you just need to refresh the web page or refresh the application, and that will re-establish the connection and re-establish the audio. So apologies in advance, it is a known problem in the software, but should only affect a very small number of people. So on with today's presentation, which is building robust pharma physical properties analysis. And your speaker today is Dr. Phil Bone. So Phil's background is in medicinal chemistry, which he studied at the University of Leeds, where he also went on to complete his PhD, which focused on the computational design and synthesis of analogues for glycopeptide antibiotics. Following that, Phil then went to work in more research-focused roles, particularly the CRO, specializing in sonocrystallization and particle engineering, and then transitioned into a more service-focused role where he specializes in systems focused on laser diffraction and image analysis. In 2015, he joined the Micromeritics team, again is focusing on a much more service support role, but in 2019 expanded his role to move into a more hybrid role where he looks after customers within the pharmaceutical sector, helping them get the most out of their investment, helping them understand their data, and understanding and supporting their applications. So with that, I will hand over to Phil, who will tell you a little bit more about that topic. Please bear with me while I just transfer to Phil.
Thanks for the intro. Hi everyone. As my introduction indicates, I do both service and applications work for Micromeritics. But before that, I was a research scientist. So far in my career, I've moved across the chemistry spectrum from what is about as organic as it can get, doing synthetic chemistry in a lab working on antivirals and antibiotics, whilst doing a little bit of computational chemistry, to what for most people is the opposite end of the spectrum, which is the physical properties of a solid. The common thread through all this, for me, well, at least the bits I liked, has been pharmaceutical chemistry. Pharma is essentially the challenge of creating a material with the right physical and chemical properties that can get to the right place and do the right thing to help a bewilderingly complex biological machine fix itself, despite said machine's best efforts to destroy and/or remove said material from the body. This is a fascinating and surprising endeavour, and quite often a testament to human persistence over common sense. The fact that we seem, as a species at least, to be getting better at this is a source of inspiration for me, and being able to help in this, at least in some small way, is what motivates me to do my job. So today I'm going to talk to you about measurements. It's not a massively surprising topic from a company who derives its name from the ancient Greek words associated with small sizes and measurement. Also, it's not a surprising topic because it's incredibly important. As if you don't measure something, there isn't a realistic way you can expect to control it. So I'd like to outline what makes a good measurement, then what I think is the best way to structure how we think about the measurements that are available to us. I'd also like to highlight some challenges where we can't measure well, and also give you some ideas about the ways these may be overcome. I'd then like to show you some bits of work I like, that I think help show the way that these physical properties are interconnected, and how they can be used to understand and control processes. So, not too much then. Right, we best get on with it. There's a lot of text on this slide. Don't worry, I'm not about to read it all out to you. It's there largely to illustrate that this isn't a simple thing, and as you read through, I'm sure you'll be familiar with these issues and the problems they present. Any of these text blocks illustrate a complex set of issues that could very well elicit the sanest response, which is to run away and find something easier to do. But for those of you that are still with me, the answer to this complex set of problems is QBD, or quality by design. But before we move on to that, I'd like to highlight some things. So the red box highlights what a measurement needs to be on a technical level: accurate, repeatable. You could add verifiable to a standard, accepted in the industry, etc. You get the idea. It needs to be fit for purpose. In the blue box, I'm trying to illustrate that a measurement needs to be valuable. So depending where you are in your campaign, it needs to either add to the understanding so you can work out the best product or the best path to it, or it needs to afford some control of the processes that turn starting materials to products. If you're measuring something and it does neither of these, you should probably stop. There's no need to throw away the knowledge you've gained by that measurement. Up until this point, it's probably given you a valuable insight into the process. It's just no longer relevant to this project. But anyway, back to QBD. So QBD focuses on the bit highlighted in green, but it answers all the other points in the process, and it's a methodology for process control that is driven by the idea of designing a robust process that delivers the desired product with a minimum analytical burden. So, having established QBD as a process control methodology, let's examine it through its use in a project. This image shows data acquired in a QBD program. It's an unusual representation of data, so I'll briefly explain the graph. Each bar represents a time period. These are not even and are anywhere from a week to a month, depending on how the data was collected, most being 2 or 3 week blocks. The earliest data is at the top, and we work down as the project progresses. The bars are centered on the vertical axis, and the wider the bar, the more data was collected. The bar width is an average of the data collected per week in that time block. As QBD is based on the principles of identifying the critical quality attributes of a product as early as possible and then creating processes that ensure the product is delivered with these properties, this results in the initial stages having a heavy analytical burden. But this creates the potential to have a lower burden later whilst retaining full process control. This shifts the analytical burden earlier in the project. The blue and the green research phases show a significant increase in the testing. This was because the properties of the components and the products were being explored, and all of the processing effects had to be quantified. These steps elucidate the design space that the processes and the materials make available. The pink process scale-up phase transitions us from measurement for understanding to measurement for control. So once the process was established at scale, unnecessary testing was cut back. During the black production phase, the excess testing was further reduced. However, it occasionally becomes necessary to revert to a testing for understanding mindset. So in the grey section, an excipient supplier had to be changed, and the new excipient had to be fully quantified. So additional testing was required. This was relatively easy, as the research testing provided a template for this. The red section shows another example of reverting to testing for understanding. Here a batch needed to be reprocessed, and this launched a kappa investigation. This was more difficult as additional parameters needed to be analysed, and this led to extra routine testing being implemented as a preventative action. At the end of the process, the analytical burden was relatively low, and significantly less than that at the outset of the project. So, good and detailed testing for understanding is the key to allow efficient testing for control. This provides a robust process that is responsive to the life cycle needs of production, but overall reduces the testing burden. I've jumped to powders here because you should measure what you make, and what you make will nearly always be either a powder or have a powder as an intermediate step. As this slide indicates, there are many powders and powder handling steps involved in creating a drug product. Therefore, powders are a critical part of any pharma manufacturing process. So, what is a powder? Well, they are more complex than most people consider, and I'll come on to that in the next slide. But I'm concentrating on powders for a little bit because they are complex. It's necessary to understand what you are measuring to know how to measure it. They're also abundant in processes and they tend to create the most issues. And if you can measure a powder well, you'll cope with any other solid form. And also, nearly all the techniques discussed for powders can be applied to any solid form. Powders are complex, and complex things cannot be both simply and accurately described. One number just doesn't tell you everything. So what do we know about the composition of a powder, and what are our challenges? Solids are often the easiest part to deal with. It's the bit of the powder that you think of when you think of a powder, the solid chunks of stuff that it's made of. Traditionally, the characterization of this can be incredibly limited, often just HPLC for compositional analysis and laser diffraction for particle size distribution. I mean, don't get me wrong, both of these are very important, but solids are more complex than just what the chemical composition and the size tell you. There are a great deal more physical properties that can be measured, and many companies and industries have already embraced enhanced solid analysis. This is one of our areas of expertise, and I'll come on to exploring these properties in the examples later. Liquids are a sticking point with powders, not least of which when they get damp and clumpy. But seriously, these are often present some of the most significant processing issues, as having the right amount of the right liquid correctly distributed through a product can often be the difference between one that works and one that doesn't. The quantity present is relatively easy to measure with techniques like Carl Fisher or GC headspace, but this does not take into account how the liquid is distributed through the powder. Gases are even more challenging, as I'm not aware of a technique used regularly to quantify the amount of gas in a powder sample, let alone its distribution through the sample itself. So some of our issues with process robustness are around liquid content. Quantity is at least measurable, but the bigger issue is liquid distribution within the powder, as this is not measurable. So I've highlighted liquids in yellow to signify that these are an area of challenge. Gases are in red, as this is an even more challenging issue, as these are hard to quantify both in terms of amount but also in distribution through the powder, and both of these can have pretty massive effects on powder performance. So, enough bad news. Solids are highlighted in green. We can measure the properties of a solid, and this is the bit we think of when we think of a powder. Therefore, most processing steps are geared up around controlling the properties and producing the right solid to give the desired performance. Understanding and measuring all solid properties can therefore greatly enhance process control. So what about powders? Well, they're in green too. We can measure these as well. Traditionally, these have not been well characterized, but they can be measured, and they really should. Controlling powder behavior is still difficult, but it can be done. So our solids and powders in green offer us points of control that allow us to deal with the challenges presented by liquids and gases. And through measuring powder properties and correlating them to the desired performance, we can produce powders with the desired characteristics. So, what are the solid properties that can begin to afford us some extra points of control? Well, the ones I've listed are a good start. I've split them into two groups. These are neither definitive, as there are other properties, nor distinct, because the two groups are interrelated. But splitting them up in this fashion groups the most strongly interrelated properties together. The first group concerns particle morphology and is predominantly the exterior features of the solid. Morphology affects powder flow, packing, compaction, and entrainment. You could add surface energy to this group. The second group, solid structure, is predominantly to do with the interior features of the solid. Dose uniformity and dissolution are governed by these, and therefore so is bioavailability. You could add pore size or pore structure and composition to this list. Both the morphology and solid structure will affect how the solid component behaves and its interactions with the liquid and gas constituents, so all of these can affect the powder performance. There are a significant number of properties here and many opportunities to control a process beyond the traditional properties of size and composition. So, enough theory for now. I'm going to show you some work that highlights these relationships and how they can be used. I'll start with solid structure, and we'll look at some AccuPick and GPick data to begin exploring this. I'm going to take you through some data from some recently published application notes, Note 180 and 181, both of which are available from our website's library section. These app notes look at how the volume, density, and porosity of some tableted products can be measured, and show how these properties might be exploited. I won't rehash the entire app note here, but as an introduction, the AccuPick measures skeletal volume by gas displacement, and it'll calculate skeletal density if the sample mass is supplied, and it'll calculate porosity if the envelope density is supplied. The GPick measures envelope volume using displacement of a quasi-solid media, and it measures envelope density if the sample mass is supplied, and it'll calculate porosity if the skeletal density is supplied. We took a range of over-the-counter tablets and measured them with both devices and generated envelope volume and density, skeletal volume and density, and porosity values for each tablet type. They were measured as multiple tablets in each sample to simulate the kind of in-process testing you might do. This is fully described in App Note 180. From this we acquired a set of data that had 5 values for each tablet type. I've plotted these on 2 graphs. The first is up and shows the porosity and volume data. The samples are arranged from left to right by ascending porosity. The graph shows that when the tablets are ordered by their porosity, there is no corresponding order in either set of volume data. So although these properties are interrelated, they are separate from each other. So if we shrink that and put it in the corner, we can pull up the density versus porosity data graph and see pretty much the same thing. The data is ordered left to right by porosity, but there is no corresponding trend in the density data. So again, although the properties are interrelated, they are separate from each other. So we have 2 tests, 5 sets of data, so far so good. But what use is it? Well, these are rapid tests and should be useful for process control. So we took the enterically coated aspirin as they are coated, and we wanted to see if this is a sensitive enough test to allow monitoring and therefore control of the coating process. Rather than testing multiple tablets per sample, we switched to testing individual tablets, and we took whole, scratched, and half tablets and measured them. The scratching and the halving is meant to simulate various amounts of damage to the coating layer or varying degrees of incomplete coating. We also took uncoated indigestion tablets to act as a control, because we expected to find little or no change in response to the differing levels of damage, as these tablets are just a single component and have no coating layer. So we generated the same kind of data sets, envelope and skeletal volume and density plus porosity, for each tablet type, and we found a lot of what we expected and a little of what we didn't. This is described in App Note 181, but I'll show you the data and I'll talk about its interpretation now. To make sense of the data, I use some difference plots, and the most interesting ones of them are shown now. So to the left-hand graph, with the bar shaded in green. This shows the change in density. The change is the comparison of each test result against the result of the whole tablet. So on the left, the whole tablet change is 0 for both tablet types. Then the scratch tablet result shows a small change for the uncoated tablet and a much larger change for the coated one. The half tablet shows higher changes compared to the scratched equivalent. But halving the uncoated tablet creates about double the change to scratching it, whereas halving the coated tablet only creates a slightly higher change than that elicited by scratching the same tablet type. The change for the coated tablet, either scratched or halved, is many multiples of the observed change for the uncoated sample. So far, so good. This is what we expected, and it's good news. It shows that looking at the density change alone would probably be a great indicator of coating integrity. And the fact that the scratch result is so high compared to the halved tablet is great, as it shows that this test is very sensitive to surface damage, as opposed to being overwhelmed if deeper damage occurs. So it should be quite discriminating. So to the second purple and blue graph. Here we're looking at the change in porosity, and the data is not quite as we expected. There is a marked change when the coated tablet is damaged, and the greater the damage, the greater the change in porosity, shown with the purple bars. This is broadly what we're expecting, but the same trend is also seen with the uncoated tablets, although to a lesser extent. This is interesting, and is almost certainly telling us something about how these tablets were formed. These are commercial tablets, and I have no prior knowledge of how they were actually made, but this data pattern strongly suggests the tablets were both made with a very similar process. Most likely a powdered or granulated mixture was loaded into a die press and then compressed into the tablet to give this distinctive pattern of a harder, well-compressed outer that is less porous than an inner portion of the tablet that is less well compressed and more porous. The aspirin will then have gone on to a second coating step. So the different response here is due to the fact that the coated tablet is coated, and so has a greater increase in porosity when the tablet is scratched, as this allows the gas to access the more porous tablet through the gap in the less porous coating. But also the aspirin is significantly smaller, so the inner, more porous core is smaller by proportion. So you don't see as big an increase in porosity when the tablet is halved. So hopefully you can see from these two graphs that there is a significant amount of data generated in these two quick and simple tests. Both graphs show data that offer insights into the tableting process, but the density change in particular offers a quite sensitive test that has the possibility to help control the coating process of a tablet. So now for something different. Well, it's not actually that different. I've come back to this slide as I want to remind you again of the interconnected nature of these properties. And also, it's a nice way to point out we've been ignoring half of them with our first example. So let's move on to morphology. I don't think anyone will find it surprising that size, shape, surface area, or any other morphological property will have an effect on performance. You may find it a little surprising that changing the properties of just one component can have a very significant effect on the performance of a formulation though. I assure you it can, and I'm going to take you through some great work published in Pharmaceutical Technology that illustrates just that. It was done by the Global Pharma Tech Laboratory of Merck, who are based in Brazil. They looked at how magnesium stearate samples from 3 different suppliers varied and how this impacted on the formulation and dissolution performance of metformin hydrochloride. The SE images shown indicate how the materials differed in morphology. The materials from suppliers 1 and 3 are agglomerates of flakes. 1 is more heavily agglomerated into rounded masses, and 3 is agglomerated into more plate-like sheets. Suppliers 2 material is not flaky, and the material is more uniform in size with a blocky appearance. First, they're characterized by SEM to give the images on the previous slide. Then they measured the surface area by BET to give the results in the table shown. Each stearate sample was then separately mixed at high or low shear rates with a metformin hydrochloride to give 6 different premixes. Each premix was then wet granulated with the addition of PVP and water as a binder, followed by fluid bed drying and milling in a conical mill. The granules were compressed in a tablet mill to give 6 different tablet types that varied only by the mag stearate used and whether the premix was made with high or low shear. The dissolution profile of each tablet type was then evaluated using apparatus 2 as described in the European Pharmacopoeia 2.9.3 using UV spectroscopy to quantify the amount of API dissolved. They found that the stearate from supplier 2, which had the lowest surface area, gave the tablets with the fastest dissolution rates. The stearates from suppliers 1 and 3 gave the remarkably similar results despite having differing surface areas. This is a little surprising, as it's expected that the dissolution of the final tablet will be slower when mixed with a magnesium stearate with a higher surface area. This is due to the fact that mag stearate is hydrophobic and it coats the API, slowing water access and therefore dissolution. The paper goes on to discuss the differing effects of shear rate, and it can be seen from the graphs that magnesium stearates from suppliers 1 and 3 are affected by the change in shear rate, and mag stearate 2 is not. They surmise this is due to mag stearates 1 and 3 being agglomerates that are more readily deagglomerated at the higher shear rates during the premix step. The higher deagglomeration results in a more complete coverage of the API with mag stearate and therefore a reduction in the dissolution rate. Mag stearate 2 is blocky and not agglomerated flakes and therefore does not show the same response. From this they conclude that particle morphology is as important as surface area when considering the final tablet dissolution properties. I agree with this completely, but I also think it shows something else that's interesting. I think if it were possible to measure the surface area of the mag stearate sample in the deagglomerated state at the premix phase without the API present, then you would probably find that the mag stearate samples from suppliers 1 and 3 had significantly higher surface areas, as the agglomerates are plates stacked face on to each other. So freeing these plates to their unagglomerated states would make the surface area higher and probably pretty much identical for these two samples. Looking at the data, I think mag stearate 2, if isolated in the same way, would be virtually the same as before the premix, because its blocky form can't deagglomerate in the same way. This is a great piece of work and nicely demonstrates how these properties are interconnected and can have real performance effects. It also leads me nicely to the concept that your measurements are much more powerful and predictive if the measurement state is comparable to the state of use. So, to our next example. This one's a short and simple case study. It's described in App Note 161, but in essence someone's trying to use lyophilization to prepare a therapeutic agent in pre-measured doses that could be easily resolved and used when required. They wanted to monitor the process by surface area, which makes sense as generally speaking the higher the surface area, the faster the material will dissolve. So if you can achieve a good and consistent surface area, you will know all your vials are very similar and should hydrate properly when they get to their site of use. Only they didn't get good and consistent results, and thus we thought we'd examine the process. We took sucrose as a model compound and freeze-dried it under a range of conditions. We collected a range of results as shown in the table. To get those results, it was necessary to break up the cake and transfer it to a sample tube. At which point it was noted that this probably altered the surface area. So there was some head scratching, and we decided to make use of an adapter so that the freeze-drying vial could be converted to a sample tube, leaving the cake intact. The adapter allows the vial with the lyophilized cake to be loaded directly to the port and measured intact. As you can see, this dramatically reduces the measured surface area. It also allows a more consistent result to be achieved by avoiding the error due to the variations in the breakup of the cake. The adapter was made available to the customer, and they were able to optimize their process. So it's clearly important to think about how you manipulate a sample in order to test it, and what effect this might have on your results. A lot of people tend to think about particle size when thinking of dissolution and overlook surface area. In fact, a great deal of the time this works, because generally speaking, particle size distribution is an inverse of surface area. If you take a material and reduce its particle size, you increase its surface area. So for the same mass of material, the smaller PSD sample will have a larger surface area, therefore it will dissolve at a faster rate. However, it's the fact that it has a higher surface area, not a lower PSD, that governs this. So now for a horrible looking equation courtesy of Noyes and Whitney. The Noyes-Whitney equation was one of the first attempts to actually quantify and model dissolution. There have been many refinements since, but all of them are still based on surface area, not PSD. So if we look at the definition of the terms, we can see that surface area is mentioned, but PSD is not. So that is why surface area was very important in our lyophilization example, because the dissolution is governed by surface area, not PSD. And also, it was impossible to measure PSD in the intact cake. The vials don't fit in the machine. So coming back to our solid property slide, hopefully you can now see the interconnected relationships within the physical properties of a solid. I'd also like to remind you again that they're even more interrelated than this picture suggests. Understanding, measuring and manipulating these properties is the beginning of controlling your production processes. This presents an opportunity to create an analytical and production process that is strongly intertwined and builds in robustness from the beginning. So let's go back to an earlier slide and remind us where the physical properties of a solid fit into our bigger picture. We've talked about solid properties in context of them being a constituent of powders, but these techniques are just as valid if your actual product is a solid. For solid products like tablets, the attributes measured by the previous examples will have an effect on the clinical performance. These are also crucial to how a powdered product will behave as well. Measuring them for powders is in many ways doubly important, because it's difficult to quantify liquid and gas content. Therefore, the most successful strategies to produce well-behaved powdered products tend to rely on controlling the properties of the solid to give the best solid-solid, solid-liquid and solid gas interactions, and then controlling the processing steps and the environmental conditions during processing to minimize uncontrolled changes in the gas or liquid content. The only way to evaluate these solid-to-solid, liquid or gas interactions is to produce a range of well-defined solids with known physical attributes, process them in a range of controlled processing steps, then measure the performance of the powder as a bulk. So this brings us to the last part of my presentation, bulk powder measurements. As powders are complex and cannot be described with only a few measurements, testing them is a process of collecting a diverse range of results that clearly define the powder's attributes. I'd like to look at this further using our Freeman FT4 powderiometer. So I'm going to take you through an example of its use and discuss the data generated and its interpretation. This is another great example. It's from my colleagues in Freeman Technology, so I may be a bit biased. But they were working in partnership with GA Pharma Systems, and this is published work. I like it as it effectively goes through the tablet production from start to finish, concentrating on QBD and looking at it from a powder perspective. The paper illustrates a path to understand tablet production and shows that through understanding and controlling the materials and the processing steps, you can generate a finished tablet with a desired hardness. As hardness is nearly always a critical quality attribute of a tablet, this is a significant bit of work. I also like it essentially repeats all the things I have previously tried to illustrate in one neat package. I'll take you through the highlights, but the reference is there, and I highly recommend reading the whole thing if you have the time. So what did they do? Well, In the first part of the paper, they use the two systems shown. Image A is the Consigma 1, which is a laboratory-scale continuous granulation and drying apparatus from GA Pharma systems. This combines a continuous high-shear granulator and a small dryer capable of running campaigns of a few hundred grams up to several kilos. Image B is the FD4 powderiometer that provides automated and comprehensive measurements of bulk powder characteristics, but it specializes in the measurement of dynamic flow properties. They use these to take two different formulations through wet granulation, then drying in a range of conditions to give granulated products. The basic flow energy, or BFE, was then measured. This showed for both formulations that altering the process conditions, screw speed, solid feed rate and water feed rate produced a wide range of granules with differing BFE results. So far, so good, but what does this tell us? I put up the graph of the data for one of the formulations, the APAP one. APAP is N-acetyl aminophenol, or Paracetamol to you and me. This shows that by changing the screw rate, or the water content, which is a function of the ratio between the solid and the liquid feed rates, you can create a wide range of granules with BFE results from as low as 1300 up to about 2700. This is great as the granules with such differing BFEs should tablet in different ways. At this point, I think we should define what basic flow energy, or BFE, is. So BFE is one of the dynamic tests of the FD4. It's a measure of how well the powder or granules move under conditions of confined flow. It's evaluated by rotating a precision blade down a helical path through a fixed volume of the powder. During this downward traverse, the torque and the axial force acting on the blade are measured, and the resistance to flow is calculated and expressed as a flow energy. So, moving on to the second half of this work, after having shown that a range of granules can be made by varying the processing parameters, the APAP formulation was granulated using the Consigma 1, again shown in image A, with 4 separate conditions shown in the table. The BFE was again measured, but this time at both the wet mass and the dried granule stage. The BFE was measured with the FD4, again shown in image B. The granules were milled and the BFE was recorded. They were then lubricated with magnesium stearate and the BFE recorded again. The resultant granular blends were then tabulated using the Modular S rotary tablet press, also from GEA, and then this is shown in image C. The milling, lubricating, and tabulating was kept the same across all the blends, so that the difference in the final tablet reflects the difference in the granule feed only. So that generated a lot of data that was neatly presented in the graph shown. You can see that the trends for each granule type tend to be maintained through the processing steps, so therefore the granular feed should affect the tablet produced. It is also obvious that conditions 1 and 2 gave granules with similar processing responses, and conditions 3 and 4 gave granules with similar processing responses. So this shows it's possible to generate a very similar granular blend from differing processing conditions. I'll shift the graph over and I'll pull up a table that shows the processing conditions again. You can see that all the conditions are in fact very different. 3 and 4 are similar only in that they have the same moisture content, and yet they give remarkably similar granules. 1 and 2 are even more surprising, as there are no obvious points of commonality, and yet they still deliver similar granules. This shows that you can tailor the granule type produced by the process, but possibly more significantly, it also suggests that it might be possible to make very similar granules from differing starting materials by carefully selecting the processing steps. This is great for process sustainability. But anyway, back to the example. Did it work? It did. The graph shown has the data from all 4 blends comparing the BFE at each stage to the hardness of the finished tablet. All 4 lines show reasonable correlation, but the wet mass and the lubricated granules are significantly lower, the wet mass having an R squared of 0.87 and the lubricated having an R squared of just 0.8. These illustrate one of my earlier points about difficult to measure components and how they have a significant effect if their distribution is uneven. The wet mass has water in it, and that is the biggest significant difference between it and the dried granules that have an R squared of 0.99. You can see from the horizontal error bars there is little to no variation in the measured BFE of the dried granules, but a significant amount in the wet mass. I've highlighted the points at the end of each line to show what I'm comparing. I suspect this is due to the water not being uniformly spread through the sample, and therefore some tested batches had more water than others. It's even more interesting that the milled and lubricated samples show a very similar trend, where the milled is highly correlating with an R squared of 0.99, and lubricated had the lowest R squared of 0.8. I've moved the highlights to show which comparison I'm making now. The difference here is the presence of mag stearate and the blending process that introduced it. I think you could probably suggest that the blend time could have been extended and the shear rate increased to lower the errors in the BFE post-blending. But that aside, it shows that these kinds of materials, water and mag stearate, that have a high effect on powder flow even at very low weight loadings, present a challenge to maintaining homogeneity. I say this because the lubricated granule BFE results also have an increased level of error. In fact, this shows the highest amount of this error. Anyway, to conclude this example, I think it's worth saying that this is a potentially powerful piece of work. It shows a strategy for exploring the design space in a production process and has illustrated that measuring one dynamic flow characteristic can control one tablet CQA. I brought you back to this slide for the final time as it highlights a couple of areas of key challenge. I hope with the examples I've illustrated the opportunities available to help overcome them. I'd also like to mention at this point one of my other favourite subjects, which is orthogonal or complementary testing. This is the idea that when you combine two or more tests of the same or very closely related properties, you can get a much enhanced understanding of the properties in question, and even sometimes the system as a whole. There is a lot to say on this and too much to add now, but I mention it as I would like to leave you with the thought that if you were to combine the enhanced solid analysis that we discussed in the earlier examples and the powder characterization demonstrated in the last example, could get you to significantly closer to a fully elucidated design space that would lead to a processing map. A processing map is a shorthand for the idea that if you know the key parameters of your starting materials and the CQAs of your product, as well as having fully mapped out how your processing steps change the material properties during production, then you can look at any batch of start material and work out the correct processing parameters to achieve the desired product. I think this is still an idealized view, and as far as I'm aware, I don't think anyone has actually achieved a processing map like this yet. I think a more realistic expectation might be that it can tell you what to do with a narrow range of starting material sets, and help you eliminate the material that will never lead to the desired product. But even at this more realistic level, this is still a significant tool for process optimization and a great step towards efficient, environmentally friendly and sustainable production. Okay, to recap, I hope I've given you an understanding of measurements, what makes a good robust one, and QBD as a structure for measurement control. Our challenges, the complexity of powders, and how gas and liquid components can present issues. And also the need to make sure the measurement state matches the state of use. The opportunities, how we can enhance measurements of solid properties, and how to fully characterize powders, and how these two things might help us overcome the issues presented by non-measurable components. And lastly, I hope I've shown you some examples that present these opportunities and how they can and have been exploited. To conclude, good analysis comes from understanding what you can measure, how and when to measure it, and how the analysis techniques work, so that you can match your analysis to what you want to achieve. This will allow you to build a robust analysis process alongside production, creating not only robust physical properties analysis, but also robust and sustainable production processes. I hope that this talk has given you some ideas of what ways you can strengthen your own analytics and your processes, and maybe even the desire to incorporate some of these techniques and technologies into your measurement regimes. With that, I'd like to thank you all for your time and your attention and ask if you have any questions. So thanks everyone, and I'll pass back over for some questions now.
Thanks, Phil. That was great. A very interesting presentation. So thanks for sharing your experiences with us. We do have a few questions, as you can probably imagine. Just before we go into those, a few people have asked whether a copy of the presentation is available. So just to clarify, today's event is being recorded, and we will send a link in the next week or so to the recorded event so that you can watch it at your leisure, or perhaps even share it with colleagues. So as I say, that will be available in the next week or so. So on to some questions then, Phil. Have I still got you? I haven't lost you in the transition, have I?
I'm still here. I hope you can hear me.
Great. Okay, thanks, Phil. So one of the first questions is, could you comment further on why surface area measurements might be preferred over particle size distribution measurements?
I guess there are two different ways of looking at that answer. So quite often PSD is used, but sometimes it fails. And the reason it might fail is when you've got an inconsistent relationship between the particle size and the surface area. So this is materials that have significant and variable internal porosity. So if you measure them before you mill them, they give you one idea of what the relationship between PSD and surface area is. But then when you break them down, when the internal porosity is higher than you might expect, you get a very large increase in surface area that you're not expecting, and when it's lower than you expect, you only get a tiny increase in surface area as you break it down. So that means you model for dissolution, if it's based on PSD, won't necessarily be as predictive as you might expect. So it's kind of like the first mechanistic reason. But one of the things is, one of the reasons why they went to surface area when they were looking at predictive models for dissolution is that there's fundamental differences in the technique. So for particle size, what you're really talking about is laser diffraction usually. So depending on which model you're using, there's some assumptions built into that. But all of the laser diffraction models assume that the materials are spherical, which they're usually not. They assume that they're smooth, and they're usually not. And they either assume that they're opaque, which they're quite often not, particularly when you get down to small particle sizes, or that they have a known refractive index. And quite often you measure materials where you don't exactly know the refractive index. So yeah, I mean, you can get better data by doing the refractive index analysis yourself for your materials, and people do that, but quite often people just put in, like, they look at the material and go, well, it's a white powder, so I'll put in the normal numbers I use for white powder. So you get errors that way. And there's a further difference because quite a lot of your surface area can be from the rough nature of the surface of the particle, which laser diffraction doesn't account for. So in surface area, there are some assumptions, but they're different. So the assumption is that the gas has a greater affinity for the solid than for itself. So that means that you will form a monolayer before you start forming multiple layers. That's nearly always true, but it is still an assumption. We also assume that the probe molecule has a fixed size, and that's generally true, but there's some argument over whether particular probe molecules will have a different size when they're associated with a surface as compared to in the gas phase. But for the nitrogen, which is normally used, it's generally accepted that the size is a fixed size, and that's agreed upon. So the assumptions are still there. In fact, there's another assumption when you're doing BET that the model that we use allows you to form infinite layers of gas molecules, and that's usually not true either. But you can form so many layers that it tends towards infinity, and you can ignore that. There are different BET models, but they're more complex and they tend not to be used normally. So when you look at all the different assumptions, the assumptions you make for surface area tend to have a lot less effect on the data. They're not as relevant. So the data is a higher quality with surface area, so you get a more direct relationship. And then, so it means that your surface area model is more generally applicable. So
Okay.
generally speaking, if you have the same surface area for the material, the contribution surface area mix to its dissolution is going to be the same regardless of the material. I mean, other factors will change, like its dissolution rate, that affect its dissolution rate, sorry, like its solubility in the solvent, relative things like that. But you don't need to redevelop the model, assuming that there's going to be a different relationship on the surface area, where because you're using PSD as a proxy, if you develop your model on PSD, every time you change your material, you've got to work out what the relationship between the measured PSD and the measured surface area will be. So you're effectively putting in an extra coefficient into the surface area dissolution equation that compensates for that individual variation for a material between its PSD and the surface area you'd expect for it. So
Okay.
there's those. Hopefully, I've answered the question with all of that.
Yeah, sure. Yeah, absolutely. Yeah. Okay, so looking at some others here. Somebody's asked, why do you think you saw a significant change in porosity for the uncoated tablet in that case study compared to the relatively minor change in density?
Yeah, I mean, that's a good question. And actually, it prompted quite a lot of head scratching because we didn't expect it. We expected to see the pattern like we saw with the density, where you had a big change in the coated tablet and a small change in the uncoated tablet. And we didn't get that. And then we looked at it and went, have we done the wrong test? Does this data not tell us what it should? Have we done something wrong? And went through everything and we didn't find anything wrong. And actually, the only thing that we found wrong was our initial assumption that you could assume that the tablet would be homogeneous, that it would be the same all the way through. So I don't know whether it came across in the slide when I was discussing it, but that was one of the things that we were trying to indicate, was that actually it was the assumption that was wrong, that we wouldn't expect the porosity change, not that there was some error in the data, because actually, if you think about the way the tablets are probably created, you would expect there to be a porosity change because the outer layer would be harder and more compressed. So when you get through that, it's easier for the gas to get into the tablet. I mean, obviously, it's much harder for it to get through the coating than it is to get through the highly compressed outer into the less compressed center. So you get a much bigger change for the coated aspirin than you would for the uncoated tablet. So I think we possibly overemphasized that we didn't see what we expected, but actually the results make a lot of sense.
Okay. Okay, so and I think probably still in that same case study, there's a couple of related questions here. So in the AccuPic and the GeoPic data, you showed a lot of bar graphs. Somebody's asked if you can just clarify the units that are used there for each of those, but also comment on the repeatability, the error values of each of those measurements.
So I was kind of expecting the error question, so I've actually got myself a little table of cheat data to help answer this one. But the bar graphs, the units, so the first set of data, you're looking at porosity, which is a percentage, and then against volume. So the volumes are actually in cubic centimeters. the second bar graph that you come across where we're comparing the density data to the porosity, the density is in grams per cubic centimeter and the porosity is still in percentage. So then when we go on to the ones where we're looking at the density change or the porosity change, actually in both cases they don't have any units because you're taking away two different values from each other, both of which are units. So it's just the difference between two values. So hopefully that makes sense of the units. The errors are more complicated, and actually they're discussed in quite a lot of detail in the white paper that's going to come out, but unfortunately it isn't out yet, so I can't just send you there to get around this. So to try and give you a bit of an idea of what the errors are, in the first part where we're talking about the actual data that comes out of the systems, so we're talking about skeletal volumes, envelope volumes, skeletal density and envelope density. The system actually reports the error values for us for them, so we've just gone with those because they're the equations for how we calculate those errors are available in the manuals. So somebody's already went to quite a lot of rigorous effort to make sure they're sensible values of error. So we're just reporting them directly. So for skeletal volume, it was 0.0031, so plus or minus that value. For envelope volume, it was 0.0113, again plus or minus that. Skeletal density is plus or minus 0.0016, and envelope density was 0.0047. So they're really small values. For the density, you should also, should put some kind of evaluation of the errors that are involved in weighing, but the machine accepts density, accepts the sample mass as a 4 decimal place number, so we used a 4 decimal place balance and put them in with that. So the error for the weight is only in the 5th decimal place. So for the purposes of the discussion today, it's not really significant. So I mean, the errors are really small compared to the values. So I mean, we're looking at.
That's great. I think that clarifies it, Phil. I think that answers that question. We've probably got time for one more quick question, coming to the end of our allotted time now, but I think it's in the next case study when you were looking at the dissolution profiles for the different magnesium stearates. You talked about the differences between magnesium stearate 2 compared to 1 and 3, but why do you think that 1 and 3 generated similar dissolution profiles despite quite significant differences in surface area?
Yeah, I mean, it's a good question. Because actually, if you look at the data, and sort of what you would expect, the higher the surface area that you measure for the mag stearate, the slower the dissolution you would expect in the end tablet. So you would expect 1 to result in a tablet that dissolves slower than 3. And yet, when you actually look at the dissolution profiles, they seem to be particularly, like, virtually identical. I mean, particularly when you look at the high shear rate ones, they seem to overlay pretty well. I mean, the biggest difference you see between 1 and 3 is the gap between the high and the low shear rate measurements is much smaller when you look at 3 than it is with 1. So this suggests that 3 breaks down more easily than 1 does. So it's actually, its behavior when it's in the low shear premix is much more similar to when it's in the high shear premix, so it breaks down quite easily even at relatively lower shear. So I think you guess that these the platelet-like tablets seem to break up a bit better. They seem to be easier to break down. And the more 3-dimensional structure of the agglomerates in 1 is harder to break down. That's why you get a bigger difference between the low shear rate and the high shear rate. Does that answer the question?
Yep. Yeah, absolutely. I think so. Yeah, that's great. Thanks for that, Phil. I think that pretty much consumes the time that we've allotted today. If any of you have raised any questions, and there's one or two that, apologies, I haven't had a chance to get to, but if you have raised a question, rest assured that one of our applications team will come back to you in the coming days. If you do want to learn more about anything Phil's talked about today or anything in the Micromeritics product portfolio, you can visit micromeritics.com or contact us via info@micromeritics.com. When you do leave today's webinar, there is a short survey at the end. We'd really appreciate it if you could just take a few minutes to complete that. It's really useful feedback for us and helps us tailor future events as well. But other than that, just like to thank everybody again for joining. Again, hope you found it valuable. Hope you found it interesting. Thank you, Phil, for your time and for your presentation. And wish everybody a good day and goodbye. Thank you.